friends to a new session on genomics and proteomics. In this session, we will discuss DNA sequencing, sequencing strategies, templates, chemistries, labeling and detection, future methods. DNA sequencing. DNA is the information stored that ultimately dictates the structure of every gene product, delineates every part of the organism. The order of the bases along DNA contains the complete set of instructions that make up the genetic inheritance. Sequencing is a key technique in genomics. The term DNA sequencing refers to methods for determining the order of the nucleotide bases adenine, guanine, cytosine and thymine in a molecule of DNA. A wide variety of DNA sequencing technologies have been developed in 1970s, resulting in gains of many orders of magnitude in speed and accuracy. The first DNA sequence was obtained by academic researchers using laboratories methods based on the two-dimensional chromatography in the early 1970s. By the development of dye-based sequencing method with automated analysis, DNA sequencing has become easier and faster. Although two different DNA sequencing methods have been developed during the same period, Sanger's dideoxy chain termination sequencing method has become the method of choice over the Maxam-Gilbert method. The DNA sequencing began when Sanger's studies of insulin first demonstrated the importance of sequence in biological macromolecules. The Sanger dideoxy method or chain terminator method, developed by Frederick Sanger and co-workers, awarded the 1980 Nobel Prize in Chemistry, has been dominant in the field. The method relies on replicating many copies of the to-be sequenced DNA segment in 4 separate in vitro reactions, where each reaction is associated with one of the 4 dideoxynucleotides. Instead of a deoxynucleotide, a dideoxynucleotide, that means the analogues of normal deoxynucleotides in which the 3'OH group or 3'hydroxyl group has been replaced by a hydrogen, is recruited, the replication then suddenly terminates at precisely that base. Hence, each reaction produces many DNA fragments of various length with the same non-base at the end of each fragment. The to-be sequenced DNA sequence can simply be read from the length distribution on the gel using gel electrophoresis. Later, developments modified this protocol to use fluorescently labeled dideoxynucleotides, which permits one reaction containing all 4 chain terminating bases rather than 4 separate reactions. Recently, next generation sequencing technologies based on parallelizing the sequencing process have made it possible to produce thousands or millions of sequences at a cost much lower than those of the traditional terminator based methods. DNA sequencing strategies, dideoxy sequencing or the Sanger's method. So-called first generation sequencing technologies which emerged in 1970s included the Maxam-Gilbert method, which was developed by Alan M. Maxam and Walter Gilbert, and the Sanger method or dideoxy method, which was developed by Frederick Sanger. In the Sanger method, which became the more commonly employed of the two approaches, DNA chains were synthesized on a template strand, but chain growth was stopped when one of the 4 possible dideoxynucleotides which lack a 3'hydroxyl group became incorporated, thereby preventing the addition of another nucleotide. A population truncated DNA molecules was produced that represented each of the sites of the particular nucleotide in the template DNA. The molecules were separated according to the size by electrophoresis, and the inferred nucleotide sequences was deduced by a computer. Later, the method was performed by using automated sequencing machines in which the truncated DNA molecules labeled with fluorescent tags were separated by size within thin glass capillaries and detected by laser excitation. The process of DNA synthesis and replication involves DNA helicase, DNA polymerase, DNA template and deoxynucleotides. DNA replication starts when DNA helicase unravels the double helix structure to a single-stranded DNA and form a replication fork. RNA primase introduces a primer that binds to the single-stranded DNA. DNA polymerase then binds to the replication fork and starts DNA synthesis by sequentially adding nucleotides to the 3' hydroxyl end of the RNA primer bound to the DNA template. The result is the creation of an extension product. The DNA sequence is copied with high fidelity because at each base on the DNA template, DNA polymerase incorporates the nucleotide that is complementary to that base. Thymine is complementary to adenine and guanine is complementary to cytosine because they can form hydrogen bonds with each other. Sequencing of the model genomes is most important application of sequencing in the last 2 decades. It consists of 2 basic steps. First, many copies of subsequences of a few hundred of base pairs or BP are generated by the sequencers. These small pieces of sequences with every base known are called as reads. Then the reads are assembled to reconstruct the genome. There are possible base calling errors in the read generation step because efficient methods or algorithms are needed to analyze image data and correctly call a base at each position. At the assembly step, there is no prior knowledge of how the reads are supposed to fit together, and furthermore, assembly is analogous to solving a massive one-dimensional jigsaw puzzle with the pathological property that many of the pieces occur many times in different places due to the repetitive nature of genomes. Automated fluorescent sequencing. Since the completion of the Human Genome Project, technological improvements and automation have increased speed and lowered cost to the point where individual genes can be sequenced routinely and can sequence well over 100,000 billion bases per year, or an entire genome can be sequenced. Since the completion of the Human Genome Project, technological improvements and automation have increased speed and lowered cost to the point where individual genes can be sequenced routinely. One new sequencing technology involves watching DNA polymerase molecules as they copy DNA, the same molecules that make new copies of DNA in our cells, with a very fast movie camera and microscope, and incorporating different colors of bright dyes, one each for the letters A, T, C and G. This method provides different and very valuable information than what's provided by the instrument systems that are in most common use. Another new technology entails the nanopore based DNA sequencing. Involves threading single DNA strands through extremely tiny pores in a membrane. DNA bases are read one at a time as they squeeze through the nanopore, and the bases are identified by measuring differences in their effect on ions and electrical current flowing through the pore. The DNA to be sequenced is prepared as a single strand or template. DNA is supplied with a mixture of all 4 normal deoxynucleotides in ample quantities. They are DATP, DGTP, DCTP, DTTP. A mixture of all 4 dideoxynucleotides, each labeled with a tag that fluoresces a different color, each present in limiting quantities. DDATP, DDGTP, DDCTP, DDTTP and DNA polymerase 1. Because all 4 normal nucleotides are present, chain elongation proceeds normally until by chance DNA polymerase inserts a dideoxynucleotide shown as colored letters instead of the normal deoxynucleotide shown as vertical lines. If the ratio of normal nucleotide to the dideoxy versions is high enough, some DNA strands will succeed in adding several hundred nucleotides before insertion of the dideoxy version halts the process. At the end of the incubation period, the fragments are separated by length from longest to shortest. The resolution is so good that a difference of one nucleotide is enough to separate that strand from next shorter and next longer strand. Each of the 4 dideoxy nucleotides fluoresces a different color when illuminated by a laser beam, and an automatic scanner provides a printout of the sequence. Maxim Gilbert's chemical degradation method. Before the popular Sanger sequencing came about, there were 2 DNA sequencing methods introduced by Alan Maxim and Walter Gilbert in 1973 and 1976. The first is known as the wandering spot analysis, which reported sequence of a whopping 24 base pairs. The second, more effective yet still limiting method using chemical sequencing. This means it used chemical processes to terminate DNA strands. These fragment DNA pieces were then run through a gel to resolve the sequence order. In this method, which is illustrated in the figure, following steps are involved. For the preparation of homogeneous DNA enzyme, restriction endonuclease is used and DNA is cut at a specific sequence. The second step, since DNA has sugar phosphate backbone, phosphate present at the 3' end of the cleaved DNA will be removed by phosphatase and replaced by radioactive phosphate using kinase. The third step, the radioactive labeled DNA fragments again treated with another restriction endonuclease and further cuts the DNA fragments. Fourth step, divide the mixture into 4 samples, each treated with a different reagent having the property of destroying either only G or only C, or A and G or T and C. The concentration of reagent is so adjusted that 50% of target base is destroyed, so that fragments of different sizes having phosphorus 32 are produced. The DNA fragments are ran through gel electrophoresis. Electrophorese each of 4 samples in 4 different lanes of gel. Fifth step, autoradiograph the gel and determine the sequence from positions of bands in 4 lanes as shown in the figure. 4 base specific chemical samples are produced. To visualize the fragments, the gel is exposed to X-ray film for autoradiography, yielding a series of dark bands, each showing the location of identical radio-labeled DNA molecules. Early strategies: shotgun sequencing and pairwise N sequencing. In the shotgun sequencing method, several copies of a DNA fragment are cut randomly into many smaller pieces. All of the segments are then sequenced using the chain sequencing method. The fragments are then analyzed with the help of a computer to see where their sequences overlap. By matching overlapping sequences at the end of each fragment, the entire DNA sequence can be reformed. A larger sequence that is assembled from overlapping shorter sequences is called a contig. From looking at the overlapping information in these 3 fragments, we can reconstruct the entire DNA sequences using shotgun sequencing. Originally, shotgun sequencing only analyzed one end of each fragment for overlaps. This was sufficient for sequencing small genomes. However, to sequence larger genomes, such as that of a human, led to the development of double-barrel shotgun sequencing, more formally known as pairwise N sequencing. In pairwise N sequencing, both ends of each fragment are analyzed for overlap. Pairwise N sequencing is therefore more cumbersome than shotgun sequencing. but it is easier to reconstruct the sequence because there is more available information. Template. Template is a very common problem area. If the chromatogram is blank, has very low signal or starts well but gradually dies out, the template should be examined. For plasmids in the 3 to 10 kilobase range, 0.2 microgram is a good amount of DNA to use. More is almost never better. It is informative to run your template on an agarose gel using some standard of non-concentration and estimate the relative fluorescent intensity following gel staining. One problem is that accurate quantification of DNA is not easy. Template composition is a factor that can cause sequencing problems. GC-rich templates will generally sequence fine unless there are particular regions of very strong secondary structure. A diagnostic trace pattern for this sort of template would be a chromatogram that looks great to a certain point and then suddenly dies out. Poly A or poly T regions will often cause difficulty. The chromatogram will look fine up to the poly T or A stretch, then earlier be very noisy, peaks, under peaks or just long rolling hills of the 4 chromatogram colors. This results from polymerase slippage on the poly T or A region and is a difficult issue to resolve. Another strategy is to design anchored primers, a poly A or poly T sequence with the final 3 base being either C, G or T or G, C and A for the poly T primer. With these primers, the poly A or T stretch needs to be at least 17 bases long and special conditions for annealing, that means the 42 degrees Celsius and cycling at 52 degrees Celsius are recommended. Sometimes this works well, but it may require the user experimenting with different conditions. Finally, di and trinucleotide repeats can cause poor data. It is usually pretty obvious when such sequences are at fault. DNA sequencing chemistries. Cycle sequencing is a simple method in which successive rounds of denaturation, annealing and extension in a thermal cycler result in linear amplification of extension products. These products are then injected into a capillary. The cycle sequencing is focused on 2 general categories of sequencing chemistry approaches: di-primer chemistry and di-terminator chemistry. Sequencing with di-primers. 4 separate reactions are performed when using di-primer based sequencing chemistries. Each reaction is labeled at its 5' end with any one of the 4 different fluorescent dyes. Each of the 4 sequencing reactions will contain fluorescently labeled either a blue, green, yellow or red labeled primer. The color of each reaction corresponds to either A, C, G or T. Dideoxyribonucleotides DDNTPs are present in each reaction mix and randomly terminate DNA synthesis, creating DNA fragments of varying length. Since a fluorescently labeled primer is used for extension, all terminated fragments are fluorescently labeled. Following a sufficient number of cycles to allow for optimal generation of extended products, the 4 reactions are combined and electrophoresed. Sequencing with di-terminators. Fluorescent DNA sequencing can also be performed using a chemistry in which the dyes are attached to the DDNTPs, thereby requiring only 1 reaction tube per sample instead of 4. Since only 1 reaction tube is required for the di-terminator reaction, this chemistry is simpler to use than di-primer chemistry. DNA template, unlabeled primer, buffer, the 4 DNTPs, the 4 fluorescently labeled DDNTPs and DNA polymerase are added to the reaction tube. Fluorescent fragments are generated by incorporation of dye-labeled DDNTPs. Each different DDNTP, that means the DDATP, DDCTP, DDGTP or DDTTP will carry a different color of dye. All terminated fragments, those ending with a DDNTP, therefore contain a dye at their 3' end. Future methods or next generation sequencing, NGS technology. Next generation or second generation sequencing technologies have largely supplanted first generation technologies. These newer approaches enable many DNA fragments, sometimes on the order of millions of fragments, to be sequenced at one time and are more cost efficient and much faster than first generation technologies. The utility of next generation technologies was improved significantly by advances in bioinformatics that allowed for increased data storage and facilitated the analysis and manipulation of very large data sets, often in the gigabase range. The RNA sequence assay, relying on the next generation sequencing instruments, provides more comprehensive information on a sample's RNA contents, known as transcriptome data. Another noteworthy example is the ChIP-seq assay, which combines next generation sequencing with ChIP or chromatin immunoprecipitation for detecting the genome-wide DNA binding sites of a protein of interest. Noise of these data comes from possible sequencing or base calling errors and the need to map the sequence segments back to the genome. Several methods have been developed to analyze these data using ad hoc approaches or some probabilistic modeling. Applications of DNA sequencing technologies. Knowledge of the sequence of a DNA segment has many uses. First, it can be used to find genes, segments of DNA that code for a specific protein or phenotype. If a region of DNA has been sequenced, it can be screened for characteristic features of genes. For example, open reading frames or ORFs, long sequences that begin with a start codon, that means the 3 adjacent nucleotides. The sequence of a codon dictates amino acid production and are uninterrupted by stop codons, which, except for one at their termination, suggest a protein coding region. Also, human genes are generally adjacent to so-called CpG island clusters of cytosine and guanine, two of the nucleotides that make up DNA. If a gene with a non-phenotype, such as a disease gene in human, is known to be in the chromosomal region sequenced, then unassigned genes in the region will become candidates for that function. Second, homologous DNA sequences of different organisms can be compared in order to plot evolutionary relationships both within and between species. Third, a gene sequence can be screened for functional regions. In order to determine the function of a gene, various domains can be identified that are common to proteins of similar function. For example, certain amino acid sequences within a gene are always found in protein that span a cell membrane. Such amino acid stretches are called transmembrane domains. If a transmembrane domain is found in a gene of unknown function, it suggests that the encoded protein is located in the cellular membrane. Other domains characterize DNA binding proteins. Several public databases of DNA sequences are available for analysis by any interested individual. Sequencing technologies also play a key role in the assays that require the identification of DNA sequences. Examples of such use include the serial analysis of gene expression and its updated version, the CAP analysis of gene expression. Both methods measure the relative frequency of different RNA transcripts in a high throughput fashion. The applications of next generation sequencing technologies are vast, owing to their relatively low cost and large scale high throughput capacity. Using these technologies, scientists have been able to rapidly sequence entire genomes, that means the whole genome sequencing of organisms, to discover genes involved in disease and to better understand genomic structure and diversity among species generally. Now let us summarize the session. DNA sequencing refers to the methods for determining the order of the nucleotide bases adenine, guanine, cytosine and thymine in a molecule of DNA. Two different DNA sequencing methods have been developed: Sanger's dideoxy chain termination sequencing method and the Maxam-Gilbert method, so-called first generation sequencing technologies. Sanger's dideoxy method relies on replicating many copies of the to-be sequenced DNA segment in 4 separate in vitro reactions, where each reaction is associated with one of the 4 dideoxynucleotides. Maxam-Gilbert method involves chemical processes to terminate DNA strands. These fragment DNA pieces were then run through a gel to resolve the sequence order. Template is a very common problem area. Template composition is a factor that can cause sequencing problems. DNA sequencing chemistries involves the cycle sequencing, which is focused on 2 general categories of sequencing chemistry approaches: di-primer chemistry and di-terminator chemistry. Next generation or second generation sequencing technologies have largely supplanted first generation technologies. The RNA-seq assay, relying on the next generation sequencing instruments, provides a more comprehensive information on a sample's RNA contents. Before we move to the next session, please try to answer the following questions. Write an overview on DNA sequencing. What defines automated fluorescent sequencing? Briefly explain the labeling and detection process in DNA sequencing. Discuss importance of next generation or second generation sequencing technologies. Discuss the applications DNA sequencing technologies. References: Introduction to Bioinformatics, Atwood and Parry Smith, Pearson Education; Bioinformatics, A Beginner's Guide by Jean-Michel Clevier, John Wiley and Sons; Structural Bioinformatics by Philip A. Bourne and Helge Wiesing, Wiley; Bioinformatics, Methods and Applications, Rastogi, S.C., Mendiratha, N. and Rastogi, P. Prentice Hall of India Private Limited, New Delhi. Hope this session was beneficial for you. We shall meet again in the next session. Until then, bye.